Okay, guys, we are ready to start. This is the session on seed orchards and seedlings, and welcome everyone. I am David Gwaze. I work for the U.S. Forest Service, and our first speaker, I think in the agenda, I know it says Claire, but Jeff DeBell is going to be giving the presentation, and Jeff is from the Washington Department of Natural Resources. Jeff?
Okay, that's on it sounds like. Yeah, so Claire was going to give this presentation, but ended up not being able to make it. But this is really a team effort, as you can see there, and there's actually a number of other people working on this. In fact, Andy was in on this from the start. And this is really about addressing seed needs, but as you know, you've all seen the reforestation pipeline thing and the acknowledgement that the first step is seed. And from this same paper, there's this graph of what people said were barriers to increasing reforestation capacity. And this one, seed's not the biggest one they cited, but up there. And that really depends on where you are. If you're in Western Washington or Oregon in lower elevation and you're planting Douglas fir, there's more than enough seed around. We all have very active seed orchards. But if you are on the east slopes of the Cascade Mountains or in southern Oregon or California and you're trying to plant ponderosa pine, it's a very different story. So that's where our project comes in. So this map here, obviously Washington and Oregon, and the darkened areas, that's the national forest lands in Washington, Oregon. And each of those red dots is a seed orchard. And you can see this slide was put together by Aaron Baumgart, but you can see that we have quite a few species represented in the system of seed orchards, and then when you consider the number of zones, it's really very large. This was really a remarkable accomplishment. Most of this done in a lot of it done in the 1980s, maybe a little bit in the 90s, a little bit earlier than that. But just a tremendous sampling of the tree species diversity and set up in places to make it easy and efficient to produce seed. For those of you who aren't familiar with the history, what happened about the time that all these orchards got established or shortly afterward, things changed drastically in the Forest Service, and what had been a very large organization with big budgets and lots of staff all shrunk just drastically in the late 90s. Funding went away, staff went away, all that sort of thing. So these orchards really got sort of left behind. Not a lot of money or time was put into them. And in some cases, actually were damaged. They came to be viewed as not important. One reason was timber harvest levels dropped precipitously. So I thought, well, if we're not cutting trees, we don't need to plant. If you don't need plant, you don't need nurseries. Some of the nurseries were shut down. And if don't need nurseries, then you don't need seed. And to the extent that there was regeneration needed, I think the idea was going to lean on natural regeneration. So Andy and anyone else can help jump in here. I don't want to speak for another organization. This is just sort of, putting together the history based on my friends in the Forest Service and what I saw over the years. But climate change has changed this perception. While some cases in the last 20 years, my colleagues have had to watch while fences are being torn down to let elk come and graze on the seed orchards, and then bears come in and kill the trees. Now I think we're starting to see some recognition of the importance of these going to the future. And so now we're working together with the Forest Service. There's many people now interested in the seed production potential of these sites. So this gives you an idea. These are all sites in eastern Washington that we're working on right now, but they can vary from the top two pictures where if you walked into there, you might just think you were in the woods. It doesn't look a lot different, all of the ingrowth and that sort of thing. Some, like the one in the lower left, are reasonably good condition, but over the years, the trees have just grown larger, and the plan would have been to thin them, and that didn't happen. Now we have crowns touching, and it's not great for seed production and is more vulnerable to loss to fire. And some are actually in not bad condition, like the one in the lower right. So we have the whole spectrum. And the same goes for the monumentation. In some cases, we have maps, good data sets. There's still tags on the trees. Other cases, we are having a real hard time finding those things so that we know just what we're working with. But this is what we're really worried about. You know, every year, orchards are threatened, particularly east of the mountains. And if we can't get in there and do something to minimize this risk, we really lose those enormous investments from several decades ago. So before I go further, one thing I wanted to point out is when we talk about climate change adaptation, you know, a lot of us are technical people and we sort of jump right to the technical kind of things that need to happen. But as we've started to work with this, I've realized increasingly that part of climate change adaptation is really administrative adaptation, changing the way we work, you know, the decision processes to allow us to do the technical things. And so some of those things are funding sources. You know, I work in a program where most of my career I've just sort of had stable funding and just manage my staff and we do our thing. You know, I'm now adapting to temporary funding, which, you know, those of you in the university world are very used to. I'm not used to that, so it's a little bit scary to get into a big project where the money might go, you know, go away in 2 years. But if we don't adapt, we're going to miss an opportunity to do a lot of really good work. And we're getting some, as many of you know, there's a lot of money coming down for different things. And so if we're really organized and ready to make use of it, we can make a big difference. Secondly, this really, really requires collaboration. Many of our organizations have been, maybe insular is too strong of a word, but you know, like in DNR, in my organization, we just kind of run our own operation. The Forest Service traditionally was that way too. But to do what we all need to do, we have to work together. And that is sometimes harder than you might think. You know, you don't realize how different the working cultures or the processes are from one organization to the next, but that's you know, what you find out when you start trying to do something across organizations. And then decision processes and authorizations can get in the way. So I'll give you 2 examples of this, one from each organization. In the Forest Service, historically, a seed orchard was controlled, and decisions about that were made by the local district silviculturist and district ranger, which was logical because that's where the seed was going to be used. Now we're in a situation where a seed orchard might be relevant to a different national forest or even a different Forest Service region if you're near those borders. And yet you have a local person that may have very little interest in that orchard making a decision that affects a different part of the land base. So that's not built very well for climate change. And similarly, on our side, we're being the state, all of our infrastructure, all our offices are located in Washington. I don't have a single employee in Oregon. Our ability to work with the Forest Service is designed to do that in Washington, yet some of the seed sources I'm very interested in are in Oregon. So we have to figure that out. So we're just not built structurally for dealing with that. Okay, so getting back to the orchards, what we're hoping to do in these orchards, we often, to help people understand, compare this to like the Firewise kind of treatments you might use around buildings, and explain to people we're doing like Firewise Zone 3. We want to separate the trees so the canopies aren't touching, so if you get fire into one tree, it doesn't carry through all the crowns. Prune those trees up, so if we get a ground fire, we're not taking it into the crowns. We want to remove concentrations of fuels. We don't want big piles of branches or slash that are going to generate a lot of heat to damage the crowns if a fire comes through. And remove ladder fuels, so those shrubs and small trees that have come in for the same reason we're pruning. We don't want to carry fire into the canopy. And this helps people kind of understand the objective. And fortunately, those principles are very compatible with what I would want to do to make an orchard productive in terms of cones and seed. So this gives you a little visual of what some of this looks like. This happens to be a Western Washington Douglas fir orchard, but you can see pretty good survival there on the aerial to the left. And then the right, you can see, those of you who are seed orchard managers know this is not what you want a seed orchard to look like, right? There's crown recession, all the crowns are touching. It'd be very hard to operate a lift in there. So this is why we're talking about thinning these out from the production side as well as the fire side. First thing we do is go in and see, well, we try to find maps, whatever data sets, whatever we can find, and then look for tags. You know, then we go in and if there's tags missing, we'll put up temporary paper tags to establish that row column structure so that then we can go in and start inventorying. We want to know, what's still there, the size of the trees, health, if there's, as you know, sometimes you go and there's a tree, but it looks like it's going to be rootstock, and so we don't want to consider that in our plans. Then we can start putting together the thinning plan. So you might get an orchard that looks like this, showing you what's still there. We can look at family composition, like the little graph on the bottom, how many trees per family, and wide variation. Some only have 1 or 2, some may have 10 or 12, depending, this all, of course, depends on the orchard. But in this example, we were at a 6 meter by 6 meter spacing, just under 270 trees per hectare, with 127 families. So plenty of diversity there from the standpoint of thinking about that. So if we go to thinning to 12 by 12 meters, because we don't get to choose our spacing either, you're at 20 by or 6 by 6, so you cut trees in those 6 meter increments. But this sounds radical to people, but actually we found documentation that this was always the intent, from that they were going to thin these things out to about this spacing. So our first, we do some little simulations and figure out if we're going to cut out every other row and column, which are the best ones to leave the most trees and the best representation. And so we can look at kind of the statistics and look at how the family composition varies and all that, with the blue being the trees that are left and the orange being the trees you're cutting. But then we can go and modify that a little tweaking, but manually, so we can keep. One of the things that's very different in this project compared to what some tree improvement folks would be interested in is we're not trying, in fact, in many cases we don't have gain data, but we're trying to maintain a nice genetic base to really maximize the representation there. And so we can do this. We add a little clumpiness to the distribution, but we're still paying attention. If we have a clump, we make sure we leave some gaps in between. But if you look at that graph on the bottom, it's much flatter. We're keeping at least a couple trees through almost all those families. This is better at meeting the Forest Service objectives. And who knows, someday maybe we want to test this. We don't know what we should keep and what we should cut. So from the standpoint of fire, we don't want as much clumpiness, but this does, it's a compromise, right? So we keep more diversity there. This is what the process looks like on the ground. We've done one of these so far all the way through. So we first send a hand crew through, prune up the trees that are going to be left and toss those branches away so we're not running equipment right up near the base of the tree. And then sometimes we just put a masticator in there, a big machine with a big, mulching head on the front. And it can take trees up to 12 inches, even larger, just chew them up. And this is the difference that you're seeing. So the upper photo, you can see this is a Douglas fir orchard where there was lots of ingrowth over the years. It was never controlled. So this is a gate, and there's a 2x4 standing right there. But you can see some of these are orchard trees, but a lot of them aren't. So we went through, marked all those up, pruned them, and then this was what it looks like afterward. This is that same 2x4. And now you can see all the way through that. Now, you'll notice that the stalking is lower than you might expect in an orchard, and a lot of that is bear damage, because this is one of those orchards where you take down the fence so elk can graze, and then the bears get in. Another photo, this is an area here where the ingrowth wasn't as large, maybe 6 to 10 feet tall, but a lot of it. You can't really see through the orchard very well, but then this is the after. So we're just on the front end of this, working with our colleagues in the Forest Service. We got almost $900,000 from our legislature. Even though this is federal land we're working on, I think everyone understands that we benefit from that. So we're this summer in the process of doing all the inventories and digging up the data sets, and we'll be able to, I think, treat a few orchards. There's a whole process with environmental assessments and things. Now, you might think that these orchards, had been logged and stumped and scraped and all that it wouldn't be a big deal, but we still do have to go through all that. So hopefully next summer we'll be actually operating. And then once we get that work done, then we start managing them as orchards. So that's another way we can work together here is on the Forest Service side, it's been 25 years. And much of the knowledge has been lost over those years. You know, retirements and people moving to other jobs, and you know, the new staff wasn't brought along. So one of the things we did in this orchard I just showed you is we had a girdling workshop this spring, just because you know, we're all used to this idea of girdling or at least in Douglas fir and in Washington, Oregon. It's just routine business for us. But you tell the average forester that, it sounds like a terrible idea, right? Because in their context, girdling is what you do when you want to kill a tree. So we had this workshop so they can see, you know, it's a different thing. They'll do this, get the hands-on experience, and then next spring we'll go back and they'll see the flowers and the pollen and just start building up that knowledge base and experience with different parts of orchard management. You know, next year we can manage the cone collection process and all that. So anyway, just on the front end of this, but it's an overwhelming project. You know, we're not starting all those 90 right now. We're focused on some of the ones in Washington, but we hope that, you know, you get in, you start getting some visibility on a project, you start showing some success, that generates, you know, more resources, and that some of our colleagues in Oregon will be able to do similar things. So.
Thank you so much. You finished on time.
Oh, good.
Okay, we've got time for 3 questions. Richard?
Are there some cases where it's a lost cause and really you either start over and keep that as a clone bank, or these things are spread out all over the place? That was the strategy back way when, in terms of consolidating some of these and putting them in place for easier management was required?
I think there's a lot of that kind of, you know, careful thinking that we need to do. I think some things are lost causes. Certainly there's different priorities in terms of seed needs. Another thing, you know, as you know, when these were put in, we were using quite conservative breeding zones. And so I think there's opportunities to say, hey, here's 3 orchards. They're all kind of representing kind of a similar environment. We have limited resources, you know, let's pick one of them. And so we have enough money to then hit the next area. So I think all those things will come into play, as well as things like some orchards we're not going to be able to operate on right away because fisheries people are concerned about hauling on the logging road without fixing culverts. So this whole thing is just a big dance of figuring out what we can work on now, what we can't work on now. And so certainly there's a big need for that kind of prioritization. For right now, I'm really focused on trying to create the momentum and get a few things done so you start generating that support. So haven't been too worried about trying to prioritize, more just about showing success and forward progress. But we definitely need, with 90, we absolutely need to get to that at some point.
So just a comment and a question. First of all, you guys are doing amazing work. I visited 16 of these orchards last year, and they're an incredible resource. And some of them definitely look way better than others where you already started this process. The question I have, though, so your orchards are so different from ours because they're usually in remote locations within the national forest. There's no irrigation system, and you are not doing active management. So, for example, you're not spraying for pests. So it's actually a heartbreaking experience when you go there early in the season and you think there's lots of cones on the trees, and then you come back in August and something like Diarrhea has just come in and decimated your crop. So is there a goal in the future, if you are going to be harvesting from a given site, is there any possibility of more active management on these sites, or will they always be remote?
What I think our goal within DNR will be, help the Forest Service get these stabilized, get them monumented, see what we have to work with. Then I think we will collect the scion and establish those populations elsewhere. It's just, on the federal land, I think the ability to deal with insects is always going to be extremely difficult, you know. And these trees are going to they're going to grow out of, you know, manageable size. Now maybe we cut some of those and we put in new grafts, but I think you have to be realistic about the rules on national forest land, and certainly we can have some good orchards there, but I think we would like to establish some on some other places where we have better ability to control insects and those things.
I have just a question about maybe you could describe a little bit more about that process of deciding sort of what trees to thin and that balance of sort
Mhm.
Like in, sometimes clones and then designs have ended up fairly close to each other, and then how did you kind of make that decision? One way or
Yeah.
another?
So, first thing, you just run there's 4 combinations of rows and columns to cut, right? You run that. You see which one leaves the most trees. And then we choose that one. And that's what this would look like. And then you can look at those stats. And then after that, I mean, it's just kind of a manual judgment call thing. This was the very first one I did, and I was trying to be really careful not to eliminate any families because, I didn't know how sensitive my colleagues in the Forest Service would be to losing some. I think all of us, if for this project, not for an orchard built for gain, but if for this project, all of us would have the preference of having plenty of space around every tree and having all the families still there, right? But as anyone who's worked in an orchard knows, that's not possible. So in this example, I was probably leaned heavier toward keeping all the families, being that this was the first one. I think my colleagues are comfortable. These are big numbers, 127 families. That's really good sampling. I think we'd probably discuss, dense areas like that. It's their orchards, right? It's not my call. But I might say, what do you guys think? If cutting some of these means 1 or 2 families we don't have anymore, would you rather have that? Or would you rather have the clump and take a risk that if we get a fire in there, it takes out all 8 or 10 of those? So, but that's, to me, that's the trade-off, and there's not a wrong or right answer. It's just kind of, but it's a very fiddly manual process aimed at, kind of flattening that. Does that answer your? Yeah, it's not a super technical process. It's just kind of.
Okay, thank you. But just one more question. On the funding, the 100,000, you said 900,000, right? That's good money anyway. Is that supposed to be spent over a certain period of time? Right?
2 years.
2 years. Will you be able to spend it?
Well, it all depends on how things go with the permissions in the Forest Service side. I think we can find the contractors. I think we can do the inventories. But there's, as you know, a lot of complications. So, yeah.
Okay, thank you very much.
Yeah.
Okay. South Korea. Hey, thank you for coming anyway.
Yes, thank you for the introduction. And yeah, as he told me, I'm Chae-Bin Lee from South Korea, and I'm a PhD student currently in Seoul National University. And before the talk, I would like to highlight that this study is a bit more focused on molecular experiments. So yeah, and I will just go through to my talk. And it's great to see you all here in Forest Genetics Conference. And actually, it's my first oral presentation. So I think I would like to talk about my current research, and I think it is related to 3 big keywords, research keywords. And first one is ex situ conservation, and second one is plant regeneration, and third one is DNA methylation. So my research topic is here, as indicated. So the presentation is constructed as follows. And firstly, I would like to explain a bit about the terminology and the species, because I assume that many of you guys are not familiar with this terminology. So Abies nephrolepis is an evergreen coniferous species which inhabits in subalpine zone in South Korea. And globally, it naturally inhabits in China and Russia as well. In Korea, it is susceptible to threats of population decline and habitat loss, so it requires in situ and ex situ conservation efforts. However, the studies on reproductive and some physiological characteristics are really poorly understood. So next is the explanation of somatic embryogenesis. So before I go further, I would like to ask, how many of you have heard of this terminology? Oh, wow, thank you so much. More than I expected yesterday. So it is a sexual reproduction process through somatic cells, dedifferentiation to callus, and redifferentiation to some somatic embryo. I will explain easily with the figure below, and many other rather explants can be used rather than the zygotic embryo, but I use this explant for my study. Plant has really interesting ability, which is called totipotency. Many of you may heard of, and it means that even with the single cell, plants can be regenerated whole. For human, if we cut our arm, I can be regenerated in a whole. So it is really interesting ability, I think. So from the somatic cell, it can be dedifferentiated to embryogenic callus. And callus means disorganized cell mass. And in the original explant, it has their identity. It was already differentiated to embryo, but it lost its identity and be embryogenic callus. So it can have many potential to be some organs or even to whole identities. So somatic embryogenesis can regenerate, redifferentiate it to somatic embryo. I hope this is clear for all of you. So it has some advantages as molecular and morphological studies of plant embryogenesis and other plants' characteristics, and as well as mass propagation and ex situ conservation, such as germplasm conservation. And for this one, embryogenic callus can be cryopreserved as well. However, studies are usually conducted to find optimal condition, how to induce this callus or how to redifferentiate to somatic embryo. So underlying mechanism at molecular level, such as embryogenic competence equation, were rarely understood. But recently, an epigenetic perspective has been proposed in relation to this plant regeneration mechanisms. And DNA methylation is one of several epigenetic modifications that can regulate. It means it can activate or repress the gene expression level without changing the DNA sequences. So when I did some experiments, I observed that callus that derived from the same explant showed different embryogenic callus competence. So it means the explant callus shared all the same DNAs, but it showed the different competence that one can be embryogenic callus to be a somatic embryo, but the other cannot be the somatic embryo. So my question was, could DNA methylation differences be the trigger for embryogenic competence? So here are two objectives of my research. First, compare DNA methylation status between embryogenic callus and non-embryogenic callus. And secondly, explore genes in that differentially methylated sites. So if I find some differentially methylation state between these two, which is second part I explored in that what does it mean? So as you can see below here, embryogenic callus has suspensor-like structure, and at the end, it can be like this somatic embryo. But compared with this non-embryogenic callus, it is friable, and as I mentioned, it has suspensor-like structures, but non-embryogenic callus showed brown and some disorganized structure. So now I would like to explain the methodology, and I will explain it quite quickly. So if you have further questions about it, please feel free to ask after the presentation and during the conferences. So for the induction of callus, zygotic embryos were used, and actually it is not the mandatory, but I prefer it because it is less contaminant explants. And explants are cultured by following procedures. And embryogenic callus and non-embryogenic callus were induced. Also, some of the non-embryogenic callus were subcultured to 5-azacitidine treated medium, and 5-azacitidine is one of the DNA methylation inhibitor. And the explant zygotic embryos were collected in 6 different stages, maturity stages for callus induction. And this photomicrograph is the status of that zygotic embryo by each stages. And unfortunately, in first stage, which was the end of the May, I think, we couldn't find any embryo, so it was only like started to pollinated and then just fertilized. So callus derived from the same explant that showed different embryogenic competence classified to 2, one to embryogenic callus and the other to non-embryogenic callus. For the analysis, 5 sets of EC and NECs were selected and DNAs were extracted. And MSAP sequencing were used to know these differentially methylated sites. So MSAP is one of the AFLP modified protocols, which is using methylation sensitive restriction enzyme. And I put the sequencing because after that AFLP protocol, NGS coupled sequencing techniques were used. So this is a brief workflow of MSAP sequencing. So here is the same DNA sequencing because EC and NEC share the same DNA as I told you. And if here is not methylated and the right one is methylated, which is methyl group, it cannot be digested by that methylation sensitive enzyme. So if we digest and then adaptyl ligate and PCR process, we cannot get in the hypermethylated sites. So if we end in this one and go directly to electrophoresis, it is the traditional MSAP. But if we add this sequencing process, for this one I've used Illumina HiSeq technology, it is called MSAP sequencing. So we can know if we both have this, assume that EC and EC, and if we get both same outputs, we know that both sites are hypomethylated. However, if one of them is, we cannot see that part, we can see that part is differentially methylated. I don't want to go further about the bioinformatics explanation, and so it is a brief workflow. And through MSAP and glucones generation, Illumina sequencing, and pre-processing, KMAR comparisons were conducted, and DMS the differentially methylated sites were designated. And the methylation states of EC and NEC were compared, and genes were annotated by BLAST in that DMS region. So now I would like to explain about the results. Firstly, for the embryogenic callus induction, 2nd and 3rd stage collected zygotic embryos showed high embryogenic competence. This results aligned with the previous studies that showed immature explant shows higher regeneration ability, especially in woody plants. It's really hard to regenerate, as you know. And embryogenic callus and non-embryogenic callus were both induced, even from the same explant. And also, conversion from non-embryogenic callus to embryogenic callus were observed by the treatment of DNA methylation inhibitor. 5 sets were selected for the analysis, and some of them are both treated with 5-azacitidine, but they show different embryogenic competence. And these are the actual photomicrograph of the real samples. And so after the DNA extraction and MSAP procedure sequencing, 50 more comparisons were conducted to compare methylation states, and each 5 sets of EC and NEC shared average 23% of hypomethylated sites, and remaining 77% showed differentially methylated status. So DMS were designated, and among them, some of DMS were annotated with +N. And 4 types of genes were annotated in DMS region. First is stress and cell division and structure, transcription, and kinase-related genes were found in that. Genes were also functionally annotated with GO term, and cellular and metabolic process were dominant for all sets. So genes related to defense mechanisms were discovered in DMS, and stress response related genes such as oxidase, alcohol, ABA, and auxin were identified. Also, genes related to cell division and structure are found in DMS. Genes related to transcription factors such as AP2ERF and NAC family were also annotated. Lastly, several receptor-like kinase that is known to be involved in plant development, immunity, were identified. My last part of the presentation, and I would like to sum up the overall results first. This is the first research on the process of Abies nephrolepis, somatic embryogenesis. So optimal sampling period for embryogenic callus induction has been identified from the study. And secondly, embryogenic and non-embryogenic callus of Abies nephrolepis that share same DNA but differ in embryogenic competence show DNA methylation differences. Also, 4 types of genes were annotated in those DMS region. In addition, transition from non-embryogenic callus to embryogenic callus was observed by the treatment of DNA methylation inhibitor. This is the significant and limitation of the studies. Compared to previous studies on DNA methylation status differences of callus, which compares some global DNA methylation level, which means 50% of cytosines are methylated, or 70% of cytosine is methylated. We can just compare with that figures. I think this study is meaningful that the study screened the entire CG methylation sites, focusing on the DNA sequences that have DNA methylation differences. Conversion from non-embryogenic callus to embryogenic callus by treatment of DNA methylation inhibitor also supports DNA methylation may have significant role in transition of the cell plasticity. However, the range of the analysis was really limited due to the lack of reference genome. It doesn't have reference genome yet. So only the gene body part could be analyzed. So we couldn't analyze the promoter or non-coding DNA and transposable elements. It has the big genome and low annotation rate, so I'm planning to do further analysis with the same data as Alice draft genome. So in other words, it is not published yet. So this is the last slide of my presentation, and based on DMS sequences, I think further studies on non-coding sequence methylation and multi-omics approaches, such as transcriptome or proteomics, will enhance the understanding of plant regeneration mechanism and role of methylation in plants. Also, I think it is really interesting to study that it is known to be immature plants is easy to be regenerated, but we don't know why yet. And I doubt that it is also related to epigenetic. So if I have chances, I would like to study this one. And lastly, not only CG methylation, CHG or CHH methylation, histone modifications, and small RNA interactions, which are related to chromatin remodeling, which is also epigenetical modification, should be studied to understand plant regeneration. So thank you for listening to my presentation, And comments and questions will be more than welcome. Thank you so much.
Okay, we have time for 2 questions. Please, start.
Thank you. Very nice talk, Chae-Bin. So, have you grown any seedlings from these, and do you see a difference between the plants where you used the methylation inhibitor and where you didn't use the methylation inhibitor?
Unfortunately, I couldn't go further to the real germination of somatic embryo. So it is for the somatic embryogenesis protocol to establish it, it is really important to start with explant to be embryogenic callus, and then it should be settled to proliferate without any some browning or other things. And then we can move further to embryogenic callus to somatic embryo. But unfortunately, I couldn't do further test so I only settled the protocol of that periodic thing in the first one. Thank you for the question.
Yeah, thank you again for the presentation. My question was, how did you obtain the explants? Like, which part of the bases you use, and do you think there's a difference between what part of trees you use to create the explants? Do you think that will have an effect on the methylation?
So you mean, the first part, I think I've understood it, but the last part, what do you mean for the methylation level?
I mean, like, for example, if you grab a bud and you use it to create a do you think that there will be a difference that if you use another part of the plant to create?
Yes. So I think it is a really good question. So for the in vitro culture, it is really important to choose which explants should be used. And in general, I have some of the experimental things, and I prefer seeds because it is protected with seed coats, so it is less contaminated in the inside one. But if we use some leaves or roots, it is already contaminated with some viruses, many microorganisms. And this tissue culture is conducted in in vitro condition in the medium, which has really good nutrients. So many microorganisms also prefer it. So it is really important to make low contamination. And bud is the really good another explant because it is also protected with some scales, But for the methylation level, I think it will be similar.
Thank you. Nice talk. Yeah. You mentioned the limitation, but you didn't mention, for example, the sequencing method that have been used. If, for example, we change for our genome discipline sequencing or the PGIS, for example, if it is limitation that for the method.
Actually, the techniques I've used, which is MSAP sequencing, it is quite robust technology, I would like to say, because it was, as I mentioned, it doesn't have any reference genome. So normally for the epigenetic studies, DNA methylation studies, we do bisulfite sequencing, which means if we do some bisulfite treatment, the methylated cytosine can be changed to uracil, the exit. So we can compare these two. However, Abies has a really large genome size, so we cannot cover all this one. So I've used MSAP sequencing because at least I can capture the CG methylation different sites. So I think it is not a limitation in my opinion, but if I use bisulfite sequencing, it would be better because I can capture not only CG methylation, I can find CHH or CHG, something like that.
Okay, that ends our session. Thank you, Chae-Bin Lee.
Thank you so much.